Scientists at the University of Texas MD Anderson Cancer Center report that a helper T cell called Th17 awakens the immune system to launch a custom-made attack against metastatic melanoma in the lungs. This preclinical research, reported online in the journal Immunity, opens possible avenues for overcoming cancer's ability to suppress or hide from the immune system. Senior author Qin Dong explains.
In this study, we address the function of a new subset of T cells called Th17 cells in tumor immunity. We know that in our past work and also the work from other groups have indicated Th17 cells to be very important players in autoimmune diseases. But the function of these cells in tumor immunity hasn't been clearly understood. And so these cells are often found in the tumor tissues from patients, human patients of a variety of solid tumors. So we wanted to address the function of these cells in tumors and whether or not they serve as a preventive role or promoting role in the development of cancers. So we utilize a mouse model for human melanomas, and we address the function of these cells by two different approaches. First, we analyze mice deficient in IL-17, which is a key pro-inflammatory molecule produced by Th17 cells. We found these mice exhibited greater susceptibility to the development of B16 melanoma, which is a mouse model of melanoma. And furthermore, we look at the other way, is to see whether or not IL-17 producing T cells, so-called Th17 cells, can provide a protective role against B16 melanoma. We found that was indeed the case, and also Natalia Martin Orozco, the person in the lab who did this work, extended to other types of cancers and found that in all cases, Th17 cells can serve a very potent and protective role in different kinds of tumors. And also she found that these cells also serve therapeutic, can provide therapeutic effect against B16 melanoma as well. So these, based on all these findings, we concluded that Th17 cells could help protect a host against the development of melanoma, at least perhaps other kinds of tumors. We believe that these cells do not kill tumors directly, but they play a major role in activation of cytotoxic T cells that can directly kill the tumors cells. So our findings have some implications in immunotherapy against cancer. So first, we need to think about, when we talk about tumor vaccines, we need to think about vaccines that can activate tumor-specific Th17 cells and sustain their function in tumor patients. Secondly, in adoptive T cell therapy, already existing in the clinics, clinicians have been using these so-called killer cells, cytotoxic T cells. They can recover these cells from tumor patients, expand these cells ex vivo, and then infuse patients with these killer cells. So our results suggest that we can also think about infuse patients with Th17 cells to help the activation and to sustain the activation of or the function of these killer cells. So Th17 cells may be considered to be co-transferred with CD8 cells in the adoptive T cell therapy to serve as a long-term protection against the development of tumors.